All right. Hi, everyone. Good morning. I hope that you are all doing well. So today, I'm going to talk about peripheral B cell responses. Before I do that, I have a couple of different administrative things to mention. So, first of all, remember the inquisitive assignment is due by 5 today. So be aware of that. You also should have received an email from me right before we got started here. I took a look at your answers to the doodle poll for review session timing, and there was an overwhelming first choice for Sunday at 7 p.m. So we will have the review session on Sunday at 7 p.m. It will be at this same Zoom link. So you've already found it. Good job. So I will see you all then. The all of the exam materials are posted. Remember, the exam is next Wednesday. If you want to meet with me, if you want to meet, I guess, today, then we can set up a Zoom meeting, or I will be back for meetings in person next week. And the other thing to mention is if you look at the schedule of upcoming topics, I am going to shift around some topics a little bit. So just so you know, on Monday, I'm going to be talking about NK cells. And I'm moving the discussion of immune response kinetics to after the exam. Okay, I think that's everything. Are there any specific questions that people have of things I forgot to tell you about? All right, seeing none, I will go ahead and start telling you about peripheral B cell responses. So
the back of the world.
We're going to be, focusing today on B cell responses in the periphery after having spent the last couple of classes thinking about T cell responses in the periphery. So remember that previously on B cells, we were looking at B cell development in the bone marrow, and we saw B cells developing through the phases that are shown here in yellow of doing VDJ recombination, doing negative selection, all that good stuff. We saw B cells were using RAG1 and RAG2. We're now going to be looking at B cell responses in the periphery. Frequently, we're really talking about the lymph node, the spleen, the Peyer's patches, or any other secondary lymphoid organ. So these are the pink organs. Importantly, we have absolutely no more RAG at any point in the B cell's life here. And now, as you can see, the B cell is searching for infection, potentially finding and attacking that infection. And so just like the peripheral T cell responses that we talked about in the past few classes, the important location for our peripheral B cell responses will be the secondary lymphoid organ. And so remember that the secondary lymphoid organ is going to contain antigen. And we talked about antigen coming in through the lymphatics. We'll see that again a little bit more today. Our secondary lymphoid organ will include T cells that have trafficked there through the HEV, and they will then go to the T cell zone, and our secondary lymphoid organ will contain B cells that have also trafficked there using really the same process the T cells did. But once the B cells get there, they will migrate to the B cell area. You can see this in broad strokes on the left and as well as on the right, where you see that high endothelial venule. You'll note that both the naive B cell and the naive T cell are coming in to that high endothelial venule. And then that naive B cell is kind of moving to a B cell area, while the T cell is moving to a T cell area and interacting with a dendritic cell presenting antigen. So I want you to remember that both of these things are kind of happening potentially at the same time, because we are going to need to see some of these events. I also mentioned to you last time that when we think about the B cell being activated, there are a couple of different ways that B cells can be activated. B cells can be activated either in a T-dependent or T-independent manner. Your textbook shows a similar figure to this in figures 9.3 and 9.4. And the basic difference is how exactly the B cell is getting its version of signal 2. Most of the time, or most of the time, at least for what we're going to be discussing, that will be coming from help from a T cell through CD40 on the B cell, binding to CD40 ligand on the T cell. The B cell could also, in some cases, get a second signal either through some kind of pattern recognition receptor or a complement receptor, which are shown on the top. Those would both be ways that we are seeing T-independent B cell activation. But when we see T-independent B cell activation, we will generally not be seeing class switch, affinity maturation, or production of immune memory. We won't be seeing the germinal center reaction that we'll be talking about later today. And so this is going to be a somewhat limited B cell activation. This is important for some types of antigen, particularly based on their chemical type, because as you see with a T-dependent antigen, we're really looking at soluble protein as our antigen because we have to be able to present some of that antigen to T cells in order to make an appropriate response. And so just to kind of give you that reminder, really for the rest of the time today, I'm going to be mentioning T-dependent B cell responses, where our B cell has to have CD40 on its surface engaged by CD40 ligand on the T cell surface so that the T cell can give the B cell help. This is definitely an older figure because this is a T follicular helper cell. And we'll see more about that later today. That is going to allow the B cell to proliferate. That's going to allow the B cell to differentiate and make either memory cells or plasma cells. So just like with T cells, where we went from kind of the naive T cell to either a helper T cell or a cytotoxic T cell, a CTL, we're going to see our B cell go from being sort of a naive B cell to being a memory cell or a plasma cell. And that B cell is also going to undergo somatic hypermutation and class switch, where we're going to see it change from that IgM producer that it was when it left the bone marrow, as well as make some changes to its complementarity determining regions, its variable region, so it can bind to antigen even better. In order to start the process of B cell activation, unsurprisingly, I think, our B cell has to encounter antigen with its B cell receptor. I know you're shocked. That seems really shocking for our B cell. And so you can see that happening here, where our B cell is binding to some antigen with its B cell receptor. That is going to start some signal transduction that really looks very similar to the signal transduction we saw with downstream of the T cell receptor. And we will also have the B cell internalize that antigen via receptor-mediated endocytosis. And that antigen will get degraded and presented on class 2. And so you can see all of that here. That B cell will start to be activated. And so you can see that when it's activated, it will internalize its antigens. It will start expressing MHC class 2, as well as some other cell surface proteins. And so you can see another view of this part of the process happening here. What you can notice is that, again, we have our naive B cell coming into the secondary lymphoid organ, like the lymph node, as this example shows, and heading to the B cell region to look for antigen. And at the same time, the naive T cell has come in and is interacting with the dendritic cell to become activated. And so now on the right-hand side of this slide, you can see the B cells that have seen antigen and have started their activation process. And you can also see the T cells that have seen antigen and that have become activated. And this is particularly going to be involving one special type of helper T cell that you saw on Wednesday, because what's going to become really important now is that these T cells that you see that were sort of getting activated at the same time as the B cells are now going to need to help those B cells in the lymph node in order to be fully activated. And so now the T cell and the B cell are going to have to interact with one another. You can see from this figure, it's actually showing things a slightly different way than your textbook is. But this will activate that B cell in order to do isotype switching and affinity maturation, particularly by leading to this structure called a germinal center that we will see. As you saw last time, this isn't necessarily a thing that we're going to often see with all helper T cell subsets. The important helper T cell subset that we're going to see in this process is the T follicular helper cell. Good news, because this is in the
That is nice, but I can spell right now.
Of the B cell. Can everyone make sure that they are muted? So you can see that we're going to be looking at this process in the follicle. And so happily we have our T follicular helper cell in the follicle. And our T follicular helper cell is going to make some cytokines as well as CD40 ligand in order to help B cell activation. And you can see this here. So what we're really going to need at this point is for our B cell, which has seen antigen and started its process of being activated, as well as degraded that antigen and presented it on MHC class 2. We'll now need to have an interaction with a T follicular helper cell where that antigen is presented to the T follicular helper cell, and the T follicular helper cell can provide help using CD40 ligand as well as cytokines. You might say, well, this seems a little bit weird because our B cells are in the B cell zone and our T cells are in the T cell zone. But in fact, once they have each seen antigen, as you can see here, and they've started to be activated, in the T cells case, they end up being fully activated. The B cell needs a little bit more help still. Those B cells and those T cells will move together to the border between their two zones where they will begin interacting. When the B cell starts to interact with the T cells, you can see that those B cells will in fact move again back towards that B cell zone So here we can kind of see this movement of those B cells back into the B cell zone. And you can also see that some of the T follicular helper cells are going to be moving back with those B cells. And the place where those activated B cells end up moving to is referred to as the germinal center. And so our B cells are going to move to the germinal center. You can see this here as well. So our B cells are eventually going to interact with a T cell and then move to the germinal center. You can see some details of the germinal center in cartoon form at the bottom here. So you can see our T cell and our B cell first interacting outside the germinal center. You can also see that then moving into the germinal center for further interactions. You can see our T cells and our B cells interacting throughout this process. So we've got B cells in general in blue on the right-hand side. We've got the T cells that are specific for a particular antigen in red, the B cells that are specific for that antigen in green. You can see they're in two different zones at the beginning, and then you can see that they will sort of move together and then kind of come into a larger cluster. People define germinal centers in slightly different ways. So sometimes we define germinal centers as the place the B cell moves to. And you might say, well, then how did the B cell know it was supposed to move there and make the germinal center? And there is one other player that is at the germinal center that is particularly important. So you will notice that we do have, and this is shown nicely here, some follicular helper T cells in the germinal center. But there is one other, like I said, player in the germinal center that was really there all along, that the B cell kind of went towards. And that is something called a follicular dendritic cell, or an FDC. FDCs are very annoying to many students in terms of their naming, because if you hear the word follicular dendritic cell, or FDC, you think, oh my gosh, it's a dendritic cell. In fact, follicular dendritic cells are not dendritic cells. They do not present antigen on MHC class 2. They're nucleated cells, so yeah, they do have to present on class 1. But you should not talk about follicular dendritic cells presenting antigen, because their function here is not about antigen presentation on MHC. They are not trying to activate T cells. You'll see them in this slide as well as the next slide. But what's happening in our lymph node is that we have antigen arriving from the lymphatics. And we've talked about this process before. So here you can see the antigens arriving in the lymphatics and just sort of moving into the lymphatic via the lymph. There are these cells, the follicular dendritic cells in the lymph node, that have tons of receptors on their surface. These include things like complement receptors. These include FC receptors. These include mannose receptors. They include all sorts of receptors. And I often think of a follicular dendritic cell as sort of being like flypaper. Just a thing that's there that's really sticky because it has so many different types of receptors that will bind to any antigen that happens to come around. So the FDCs are these structures, these cells that are just picking up and sort of not picking up by phagocytosis, but getting antigens stuck all over them. So the free antigen is going to enter the lymph node and bind to those follicular dendritic cells. You can also see those follicular dendritic cells here. Like I said, they are sadly not dendritic cells. They are not of the myeloid lineage. They're not even from the hematopoietic stem cell. They're something that's totally not an immune cell. But they have a ton of receptors on them. They were originally named follicular dendritic cells because under a microscope, they look kind of like a dendritic cell, which you can see on the right, where you can see an image of them. But really, they're not phagocytic. They're not doing any of our dendritic cell stuff. They just have all these receptors on their surface so they can get covered with antigen. They have a huge surface area, as you can see. And so you can kind of think of them as almost like the antigen depot. And if we have free antigen coming into the lymph node, it's getting stuck all over the follicular dendritic cell. And so that's kind of, like I said, the antigen library almost that you can think of for the B cell. It's actually really an interesting question in infectious disease in terms of how long does the antigen stay on the FDC? Are there antigens there for really long times? Anyway, so the place that the B cell is going to be moving to make its germinal center is to a location where there is an FDC with a lot of antigen. And it will start the germinal center reaction as you can see here. Germinal centers are easily detectable in secondary lymphoid organs like lymph nodes. Depending on our timing at the end, I may show you some germinal centers from normal individuals versus COVID patients that have been shown in the literature. And you can see the germinal centers really easily under a microscope. So you can see a germinal center in 3 different types of microscopy on this slide, as well as in a cartoon form. You'll notice that we've got our follicular dendritic cells, we've got our T cells, and we have lots of B cells, and we are firmly in the B cell zone. And you can see that there are different areas of the germinal center. In particular, there is an area known as the light zone and an area known as the dark zone. They are distinguishable by microscopy. You can note that the dark zone mostly has the cells that are colored green here, which are largely those B cells. The light zone has some T cells, it has some B cells, it has some follicular dendritic cells. It's got more different cell types. You can also notice that the dark zone has a larger density of cells than does the light zone. That's actually why it's called the dark zone. It's so packed with cells that the light can't go through. And so it looks darker under a microscope, where the light zone has a smaller density of cells, so light can go through more easily. And if you think back to some of the things we talked about with the thymus, that might give you a hint as to which one of these involves proliferation, because we talked about different areas having different densities of cells and how that related to proliferation when we talked about the thymus. So the germinal center is a very important part of B cell activation and looking at a peripheral B cell response. And when we are thinking about the germinal centers, we will see B cells going through the germinal center reaction. And so the way that this will work in our germinal center is first our B cell will start to become activated. It will move to the T cell zone to interact with a T cell. And then once it has interacted with the T cell, you can see it entering into the germinal center. And the first thing that happens to that B cell is that it's entering into the dark zone. And that B cell is going to proliferate. It got a happy pat on the head signal from a T cell that it was useful and its antigen was around. And so that cell is now going to start to proliferate and make many copies of itself. However, that cell is going to also do one other thing here, which is that that cell is going to turn on an enzyme, who we'll talk about a little bit later. And that enzyme is going to start to make some changes to the variable region of the B cell. Now, I want you to remember that we aren't looking at one activated B cell at this point. We're looking at a bunch of them because that B cell has proliferated. So we've got many progeny B cells. In this case, we'll imagine that B cell has divided, and we have 100 progeny B cells. Probably has not divided that many times at this point, but we got a lot of progeny B cells. And there, we're going to in each of those, we'll turn on this enzyme that is going to lead to some mutations in each of those different progeny B cells' variable regions. And that is known as somatic hypermutation. So this is a body cell, so we've got somatic. We're going to add some mutations, and we're going to add a lot of them. So it's hypermutation. And if we look at where those mutations are really going to all be centered right in the either VJ region of the light chain or the VDJ region of the heavy chain. So right in the variable region, right where we're going to see antigen binding. We're going to pick up a whole bunch of point mutations. You can see the acquisition of point mutations as well on the right of this slide. And what that will do is that will change the variable region, and that will change the antigen binding site of the antibody. And so instead of the antigen binding site that we see at the beginning, we will see a new, potentially better binding of this antibody. We'll say it's a new, better binding. You will see better binding overall in the response. And so we're going to have higher affinity. The affinity has improved or adapted, because this is adaptive immunity. And so we sometimes refer to this as the affinity maturation, the idea that the affinity has matured or gotten better. So affinity maturation is what happens to the antibody as a result of somatic hypermutation happening to the gene. But there is a little bit of a trick here, sort of an assumption that I have made in this process. We are getting a whole bunch of point mutations in our variable region. And here I tell you that overall the affinity is going to improve. But you might say, well, how are we sure that the affinity is going to improve? What if we make point mutations and we destroy the variable region or make the affinity worse? How do you know that these point mutations you're telling us about are actually improving the affinity? And that is the sort of goal of the germinal center reaction. And so what happens with our B cell is that B cell is going to acquire some mutations, and then that B cell is going to retest itself against antigen, particularly antigen on the follicular dendritic cell. If that cell does not bind to the follicular dendritic cell, then we see this process that's happening on the left, where that cell gets no further signals, no more pats on the head, and that cell undergoes apoptosis. So if that cell has destroyed its variable region, then it dies. And if that cell has good binding to antigen on the FDC, as well as binding and getting help from a follicular helper T cell, that cell is going to get a signal and be able to kind of further bind. Remember that we had that proliferation of the cells. And so there were many progeny. We made up the number of 100. Those 100 progeny are each going to make slightly different mutations. And so now we're going to have 100 different versions of the original variable region. And those 100 different progeny cells are going to be competing to try to get a signal from the follicular dendritic cell and the helper T cell. And so the ones that have made higher affinity or better affinity are going to be really good competitors. They're going to bind well and kind of crowd out everybody else. And they are going to be the ones that get the signal. And so overall, we're going to have an improvement of the response, even though each individual B cell might succeed or fail. If we actually look at the population as a whole, we're going to be getting better and better affinities. And this is sort of something you can think of as almost like natural selection or Darwinian selection on the level of cells instead of on the level of whole organisms. And so you can sort of see this process happening here. Our B cell that has started its activation process will move to the germinal center. It will both proliferate but also start doing somatic hypermutation. One thing to point out, and you'll see this more later, is that somatic hypermutation does not include the use of RAG1 and RAG2. So this is totally different enzymes. We are never seeing RAG1 and RAG2 again in the life of this B cell. So now we'll have lots of progeny B cells, and those progeny B cells will now test again with a helper T cell that's in the germinal center and a follicular dendritic cell. If we get bad signal or no signal, we'll undergo apoptosis. If we get a good signal, then that cell is going to continue to get signals. It's going to continue to go through this process. It's going to start differentiating, and it also may actually go back into the dark zone, proliferate, and mutate again. And so it may go through multiple rounds of this process in order to really improve the affinity very, very well. At the conference where I am right now, I heard a talk about some germinal centers in a particular situation, and the investigator was able to identify B cells that had undergone 750 rounds of division in a germinal center reaction. So it's pretty impressive. We don't actually know what signal tells a B cell, okay, leave versus stay, but oftentimes we'll see multiple rounds through the germinal center before we end up with our finished product cells, the plasma cell or the memory cell. There's one other kind of thing that we can think of that is a result of this germinal center reaction. So here you can see our resting B cell. Remember that our B cells, when they came out of the bone marrow, were producing IgM and IgD, and that's it in terms of isotypes. We're going to see antigen potentially in the lymph node. We're going to start to activate and get proliferation. And then we will see our affinity maturation. We will see differentiation to a plasma cell or a memory cell. But we also, in this process, see class switching. And so our B cell also may switch from making IgM and IgD, as it was doing at the beginning of its life, to making one of the other isotypes, like G, E, or A. We're pretty sure that this happens sort of as a reaction sometime in the dark zone, but the exact details of exactly when it happens relative to some of these other events are unclear. And so remember that we had those mu and delta constant regions for making IgM and IgD really close to the VDJ region. And then all of the other constant regions much further away downstream, many kb away in the DNA. And so in order to have the cell produce one of those other types of constant regions, we need to delete this intervening DNA in order to make the constant region that we choose. This happens through a process known as class switch recombination. We find upstream of each of our constant region areas of the genome something called a switch site. So you can see the switch sites listed here. This doesn't show a switch site before IgG, but there actually is one. And what will happen is we have this separate process of recombination where we'll have the cell will basically do recombination between its IgM switch site and the switch site in front of one of the other constant regions, deleting the DNA in between. So now you can see here's my deleted DNA. Here is the VDJ that used to be next to IgM. It's now next to the IgG1 constant region. And now we've thrown out the intervening DNA. So we used to be able to make M and D. We now are going to make just, say, IgG1 because we've put that constant region in close proximity to the VDJ. And we've done this by a permanent change of the DNA. Part of this has to do with which of the switch sites are available to the enzyme that does this VDJ recombination process. And the availability, the chromatin availability of the switch sites will control which type of antibody the B cell will switch to. The way that we control which one to switch to is based on which cytokine the B cell receives. So the B cell is going to see different cytokines from T cells during its activation. So if there is a Th1 response going on in the area, that B cell is probably going to see some Th1 cytokines. If there is a Th2 response going on in the area, the B cell is going to see some Th2 cytokines like IL-4. And if that cell sees IL-4, that's going to push it to making more IgG1, IgG4, IgE. If we see, say, I guess of the ones that are shown here, IL-15 is a more Th1-like cytokine. Now we're going to see, and I don't actually believe that from what it says here. In any case, we're going to see different isotypes based on which cytokine is present. That's actually going to lead to availability of different switch sites for our enzyme. There are also patients who have a disease known as hyper-IgM syndrome. And hyper-IgM syndrome is a disease where the patients don't have germinal centers. And so you can see a normal lymph node on the left, and you can see how easily visible the germinal centers are under a microscope. There are patients who don't make germinal centers, and because they don't make germinal centers, their B cells can never class switch. They only ever make that early IgM. They don't ever switch to other isotypes. And so they have too much IgM, hyper-IgM syndrome, as well as too little of all of the other isotypes, aka none of them. And in general, there are three famous genes that could be mutated in a patient to give that patient hyper-IgM. One of them is CD40 on the B cell, so the B cell cannot receive help from the T cell. One of them is CD40 ligand from the T cell, so it can't provide the help to the B cell. And the other is the enzyme that we're going to talk about that is important for this process as we go forward. And so all of these things are sort of the downstream results of the germinal center reaction. So we're going to see our B cell be activated, expand, and then class switch, perform affinity maturation, and differentiate into one of two types of cells, shown here as either a memory cell or a plasma cell. The memory cells, you can see some information about this here. So on the left or on the right, we've got our T follicular helper cell interacting with our B cell. You can see our T follicular helper cell is providing some cytokines. It's also providing CD40 ligand to interact with CD40. Remember, that interaction was key, and if either of those were missing, we didn't get the class switch and we had hyper IgM syndrome. And our B cell will start to differentiate to either a plasma cell or a memory cell. A memory cell is going to live for a long time. And it is going to make a better response later if it sees antigen a second time. And we are going to talk much more about aspects of immune memory after the exam. Alternatively, that cell can differentiate into a plasma cell. And you can see an electron microscopy image of a plasma cell on the left. You can see that it basically is a nucleus and ER, specifically as is labeled here, rough ER. This cell has a super weird, super specialized structure. If you look at the beginning of chapter 9 of your textbook, you also see an image of a plasma cell. And plasma cells have one job. Plasma cells are very, very, very specialized to do their one job. In fact, they lose every other kind of ability of a B cell. And the one job of a plasma cell is to make and secrete antibodies. That's why all they have is rough ER to make secreted proteins, particularly secreted antibodies. And so you can see this here. A naive B cell has B cell receptor on its surface. It has MHC class II on its surface. It has the ability to grow. It has the ability to do somatic hypermutation. It has the ability to class switch. It doesn't make a ton of secreted antibody. A plasma cell makes secreted antibody. And that's it. You can see that it has no for all of these other things. It stops making B cell receptor on its surface. It stops presenting on class II. It's not going to grow. It's not going to mutate. It's not going to switch. It's just going to produce all the antibodies. And that's how we are getting antibodies at a very, very high concentration. So again, if we were looking at this exact topic when I learned it as an undergrad, this is kind of where certain parts of it would have stopped. And I would have said, somatic hypermutation and class switch happen by magic, yay. But we have since actually figured out the enzyme that is involved in these processes, and we understand how these processes work. One thing that's really great about thinking about the enzyme and these processes is we have learned it's the same enzyme that does both somatic hypermutation and class switch recombination. And so this is one enzyme that has to get turned on during the germinal center reaction, and it will lead to both of these processes. This enzyme is shown here, and this enzyme is known as AID. AID stands for activation-induced cytidine deaminase. When you look at that, I know you want it to be AICD, but it's not. AICD is actually an acronym used for something else in immunology that we haven't seen yet. So sadly, it was taken when this enzyme was described. So this enzyme became known as AID. And its name really tells you what it does. So first of all, you can see that it only is turned on in the B cell when the B cell is activated because it's activation induced. So we don't see it in the naive B cell We only see it in the activated B cell in the germinal center. And this enzyme works on cytidine. Cytidine is one of our base pairs or our bases. Remember C, cytidine in DNA. You can see a structure of cytidine on the left. And what AID does is it induces a chemical reaction on cytidine to remove this NH2 or amine group and result in it being replaced with this oxygen. And so when we're removing the amine group, we are deaminating the cytidine. So in fact, this is cytidine deaminase, the activation-induced cytidine deaminase. When cytidine is deaminated, as is shown in this chemical reaction, it makes a specific product. I want you to remember that this AID enzyme is acting on DNA. This is acting on the genomic DNA. As you'll see, it's either acting on the genomic DNA in the variable region or it's acting on the genomic DNA in the switch site. It's deaminating the cytidines there and giving us the product of cytidine deamination. And the product of cytidine deamination is uracil. Shown here is uridine. And so this is a little bit of a problem. This should ring some alarm bells for you. Why is it a problem that, or what's the issue with AID deaminating these cytidines into deoxyuridine or uracil? But calling it deoxyuridine kind of helps maybe give you a hint. What biomolecule are we currently in? Are we currently doing processes with? Michael?
Or in DNA, but normally uridine's only found in RNA.
Exactly. So we're in DNA, but uracil, uridine, is only found in RNA, and we've just turned a base into a uracil or a uridine in the DNA. That's weird. If you go online and look at some memes, you will find a meme about Chemistry Cat who says, if I had to be a nucleotide, I would be RNA because it has U in it. And because RNA is the only one that's supposed to have U, and we just put a U in DNA. Bad. And so what you can see is that AID is going to act on a C in the DNA, and it's going to result in a uracil being there in the DNA. And you can see that here. And this is really bad because there's not supposed to be uracil in DNA. It's also really bad if you look at this because the uracil is paired with a G, because originally this was a CG base pair. So now you have a UG base pair. Does U go with G? No, what does U go with?
T.
Close. What does it go with, Michael? I saw your hand as well. Or Carney, what does it go with?
It goes with A.
It goes with A. It replaces T to go with A. So this is just all wrong. And so we have to do something about this uracil. And there are a couple of different things we can do about this uracil. Sometimes we might replicate that DNA. And if we replicate that DNA, then one of the strands, the strand that's GA, will just replicate and everything will be fine and we'll get back to the beginning and no one cares. And those are sad, not fun, not somatically hypermutated B cells. So we don't really care about those. sometimes will just replicate through, and when we replicate, the U will get paired with an A. And so now we'll have changed what was a CG base pair to become a TA base pair. And so we'll have made a point mutation in our DNA. Other times we will we'll actually recognize that the uracil is a problem before we replicate, and we'll do some DNA repair, and we'll remove that U. And what generally ends up happening in that case is that the repair processes that will happen just put in a random base pair. So again, we're going to get a point mutation, but now it doesn't have to be a point mutation to T. It could actually be a point mutation to any base pair here. If there were multiple, if there were sort of a more challenging situation here, for example, if we had multiple U's near each other, we might have to do a different type of repair. But if we look at the issues on sort of shown here on the left, we can see a bunch of ways that AID can lead to point mutations. So different C's in the DNA can get changed to other things. This is affinity maturation or somatic hypermutation. So basically what's happening here is AID is acting on the variable region. Particularly, it's acting against all of the cytidines in the variable region and leading to point mutations. And that's allowing us to change the variable region and potentially get higher affinity in those individual B cells. And so if you actually look very closely, you will see even all of those point mutations are really largely in the cytidines or Cs in the complementary determining region, the CDRs. So we are actually going to really change how we bind antigen and get much better interactions. As I mentioned, we can also see another option for the resolution of our uracil. So one other thing that we can have happen is sometimes we'll actually have to have the entire base removed. This is particularly important in a situation where maybe we had two cytidines, or maybe we had like a whole string of cytidines next to each other. Here are all my cytidines next to each other. They all got deaminated, so they all went to uracil. And now we're going to have a big gap because we removed a whole bunch of bases. And at this point, we need to do some kind of more serious repair on this gap. And that serious repair ends up being homologous recombination, where the cell will try to find similar DNA sequence and perform a recombination event to fix this dramatic problem. That process is what gives us class switching. And so when AID leads to those more dramatic types of changes, we will end up getting class switching. So basically what is happening in these cases is that AID is putting a lot of mutations into the IgM switch site and into some other switch site. Whichever one is available because of chromatin changes due to whatever cytokine the B cell has received. Now we've got these two sort of pieces of DNA that are going to undergo general DNA repair. They're going to undergo recombination. We're going to throw out the DNA in between, and now we're going to get class switch to this particular site. And so both of these processes of class switch recombination and of somatic hypermutation are happening because of this same enzyme AID being turned on in the germinal center to lead to this process. Yesterday, as I mentioned, I saw a presentation by someone who was talking about germinal centers. And he gave a fabulous talk about germinal centers. And he also had some really fabulous overview slides of the germinal center reaction. And he was nice enough to send them to me. So now I want to show you these overviews of the germinal center reaction that we've just seen. So let's hope this works. All right, so now what you're seeing, ooh, stop. Okay, to start out with, we've got our T cell in the secondary lymphoid organ. It is interacting with the dendritic cell. It's getting activated. Now that T cell is going to become a T follicular helper cell. It also might not, and we don't care about the cell that doesn't. There's our T follicular helper cell. We know it's a T follicular helper cell because it is making that unique cytokine, IL-21, and it is making that specific transcription factor, BCL6. It also is now, because it's activated, making CD40 ligand on its surface. That cell is going to move to the border of the T cell B cell zone, where it's going to interact with our B cell. The B cell is going to have started its activation process by internalizing antigen. It's going to be presenting that antigen on MHC class 2. We're going to have CD40 ligand on the T cell interacting with CD40 on the B cell. There's our exciting B cell. The T cell is going to be activated by that process, but so is the B cell. And so the B cells might make a plasma cell that could leave the germinal center. You can see that B cell is also going to interact with a follicular dendritic cell and test its antigen. If it tests positively, it's going to go back to the dark zone and proliferate. And then it might move back to the light zone, try again, and eventually become either a plasma cell or a memory cell. And I don't remember what happens. Oh, and our T cells are also going to be activated to memory cells here, but we care about that later. So I thought that was a pretty awesome animation, and I was excited that he sent it. You can also see our T cell being activated with the dendritic cell in the T cell zone. Our B cell, and then eventually our T cell and our B cell are going to interact. You can then see our germinal center forming with a follicular dendritic cell, T cells and B cells. And that's going to allow our B cells to differentiate. Eventually, we're either going to have memory B cells or plasma B cells leaving. All of this is happening in the B cell follicle. And the other really nice thing that I want to point out about what we see here is that there are some B cells that could get activated, go to the border between the B cell and the T cell zone, be looking out for their T cell, and if they don't find a T cell, if they don't find their matching T cell, that stops this whole process in its tracks. And so if there was a B cell that could not find its matching T cell, that B cell is never going to go into a germinal center, finish this process, make memory or plasma B cells, etc., etc. That B cell is going to just be real sad. And in fact, sometimes it's referred to as a helpless B cell. So you can see the helpless B cells nicely in this figure as well, and they will come up later on. So a couple of cool things that I want to tell you about. My guess is I'm going to tell you about the cool things, and I'm not going to get into talking about linked recognition and the problem that I wanted to go over with linked recognition, but we'll do that on Monday. because this is really cool stuff about germinal centers I just wanted to show you. So first of all, this is a paper that looks at germinal centers in people. One important thing to note is that these are actually images from the sections of lymph nodes that were taken from people at autopsy. And so you can see B cells with CD19. You can see T cells. You can see BCL6 showing us the follicular helpers. And you can see this beautiful germinal center. BCL6 is also labeling some of our B cells. And so you can see these beautiful germinal centers. And this is what germinal centers look like. And I was so impressed when I saw this paper. And this paper was actually really interesting in terms of thinking about germinal centers and COVID. So what they also did is they also looked at germinal centers from lymph nodes of COVID patients on autopsy. These were very severe COVID patients who died of COVID. And when they looked, this is what they saw. And so you can see that, in fact, infection with SARS-CoV-2 seems to completely destroy germinal center structure. So yes, there are some B cells that are turned on, but those B cells are not great. They're turned on incorrectly. And so a lot of parts of what we see in their B cell responses may be aberrant. This is actually related to problems in making follicular helper T cells, which is all related to problems of the innate immune stimulation those T cells got. There's one other thing that we have really learned about germinal center responses. To be honest, in the case of the past couple of years, we have probably learned more immunology than we had in the previous 50 in a lot of ways, because we have so many patients with the same antigens that are being examined by so many people with such fancy technology that we are learning really, really cool things. And one of the places where I've been really excited about what we've learned has been with regard to some things that I've sort of come to realize about the germinal center and the germinal center reaction. And so these data are part of sort of what we've seen. And so first, I'm going to show you part of this figure. Here, researchers were looking at the amounts of IgG that was produced in people who had gotten just the first shot of an mRNA-based COVID vaccine. People who got a second shot, either at 1.3 months after they're getting the shot or at 5 months, and people who got a third shot. And I will tell you that this whole set of experiments was done with the original strain vaccine. This was not done with the new Omicron-specific vaccine. This is all original vaccine. And so what you can see is that when you get when you get your first dose, you start making some IgG. You've class switched. You've had plasma cell formation. It's the only way you could make this many antibodies. You get a second dose, you make more. Shocker. Those go down over time, as they should, and as we'll talk about more when we talk about kinetics. And you get a third dose, oh my gosh, they go up again. All looks good. All looks exactly as we might expect. This is showing us both antibody levels on the left and the same data in terms of neutralizing titers. So how many of those antibodies are neutralizing on the right. And if we look at those antibodies over time, we can actually look to see how many of them are sort of genetically different, how many different clones there are. You can see at the beginning, in some cases there were like 12 or 71. And if you look at the pie charts, you can kind of see some differences in sequences. And as we go further after vaccination, we get more and more diverse antibodies because those antibodies have undergone somatic hypermutation. They've done the germinal center reaction like we've seen. We can actually even look and see how much mutation, how many point mutations do we have And if we look after the second dose versus the third dose, we see a difference where there's been an increase in number of mutations. So that's cool, right? Like the immunology I'm telling you about really happens. But if we actually look carefully at those experiments, what we can see is that when people get that third dose, and so here notice, say, Vax2 at 1 month and Vax3 at 1 month, all of those people got vaccinated against the original version of the virus, the Wuhan HU1 strain. Yet, as we go on, after the third dose, people are starting to make antibodies that can bind to different variants. You can see that even though this is not the Omicron-specific vaccine, we're getting antibodies against Omicron with a third dose. And this is showing on the right. So we're just seeing titers or antibody levels on the right. On the left, we can actually see binding strength. And so we have, or inhibitory concentration. So we've actually decreased the inhibitory concentration, which means that we have increased binding strength against Omicron. So as we pick up these changes, it's almost like the response is sort of anticipating variants and also making such crazy affinity that even if the virus tries to mutate away, even if the virus mutates in a way that gives you, a 10th as good an antibody response, you have such an amazing antibody doesn't care. And so we have really noticed how much this germinal center reaction is important for diversification of immune responses. And that has been a huge and exciting thing that we have learned about the germinal center response during this process. So as I said, we'll have a review session on Friday at 7, on Sunday at 7. And we'll be talking about NK cells on Monday. And remember the inquisitive assignment. And just the one last thing that I will tell you, let me make this go away, is just so you know, this is what it currently looks like outside in Hawaii at 7 a.m. So you can see kind of what we're looking at here. So I will see you guys on Sunday at the review session and then on class on Monday.
Thank you.
Yeah.
Enjoy your time.
Thanks.